Thank you, Richard. Thanks, everyone, for being here. It's great to be. I've never been to Pasadena before, so it's really nice to be here. So yeah, I'm going to give sort of, I guess, a broad framing talk based on some research that my group has been doing over the last few years. And what I'm going to encourage us to do this morning is to really think about containment in a broad sense. So I think probably broader than the often sort of default technological containment approach that we take. And I'm going to encourage us to think about the idea of social containment. The work that I'm going to share with you this morning is grounded in a report that my research team put out earlier this year. There's a QR code there for anybody. I think it should take you to a link to download the report. I also do have a few hard copies with me if you're somebody who likes to engage with paper. So just come and find me and I can give you a copy of it. But to do a little bit of just context setting, I think Richard actually set this up, so I'm not going to spend much time on this. But researchers have been thinking about possible uses for engineered organisms outside the lab since the beginning of molecular biology. And I think it's actually really interesting that the first ever US patent on a living organism was actually for a genetically engineered bacteria designed to clean up oil spills, so the famous Chakrabarty patent. Now, this particular organism never left the lab, never made it to market, but it did signal from very early on this interest in using engineered microbes in contexts outside the lab or outside of a controlled sort of biomanufacturing space. I think probably Steve Evans will share with us a little bit more about the history, but there was sort of a lacuna in releasing genetically modified organisms aside from GM crops in the US. So through the sort of late 80s, 90s, 2000s, we've seen a lot of GM crops. We haven't seen a lot of releases of other engineered organisms. It does seem, though, that this may be starting to change. So in recent years, we've seen certainly projects devoted to environmental restoration. We've seen plans for releasing genetically engineered insects for public health purposes. And we've also seen growing interest in, as Richard said, these sort of direct-to-consumer products that in a number of different arenas. So the Z-biotics might fall into sort of consumer wellness, even recreational space, right? So there seems to be the kind of change, the sort of ease of using these technologies is leading perhaps to a resurgence of ideas among researchers for how we might use genetically engineered organisms outside in sort of deliberate release contexts. So I'm a social scientist. I've spent the better part of 2 decades studying and working alongside the synthetic biology community. And the motivation behind the project that I'm going to share with you is really sort of hearing this growing interest within the research community for trying to think through, can we use biologically, can we engineer organisms for bioremediation, for conservation, for all kinds of, kind of application spaces that we haven't really tested before. But as Richard pointed out, some of these involve much more open contexts, right? Moving into that from factory to field to farm to field. We're moving into these spaces that are much less controlled, much more open. And the scientific community is very much aware of this, understands that there may be concerns, and is trying to think through how we might manage these organisms. And one of the default kind of responses that you sort of hear within the synthetic biology community is, well, we'll just build containment into it, right? We'll engineer kill switches, auxotrophy, we've got all kinds, there's a lot of effort to try and develop technological containment mechanisms as a sort of answer perhaps to concerns or questions around safety, around consumer acceptance, around public trust, and so on. So the work that we did was really sort of motivated by this almost sort of this kind of question of, well, is technological containment the answer, right? Is this the thing that's going to promote safety, trust, acceptance, the flourishing of these organisms in society? And we wanted to see basically what we could learn from what's already out there. So learn from historical cases, learn from organisms under development, and really try and understand how particular products came to market or failed to come to market. So we looked at 11 case studies. So the report that you can download has 11 case studies that are each about 4 pages. We looked at a variety of plants, microbes, and insects and animals. About half of the cases that we looked at are genetically engineered organisms that were commercialized. I should say the Aqua Advantage salmon case was not designed for deliberate release, but it's a very interesting case for many reasons, and so we did include it. We looked at some cases that were never commercialized or never made it to market. We looked at some cases like gene-drive rodents, American chestnut tree, that have yet to be commercialized, just to see what we could learn. This is another way of showing you the sort of timeline. So looking at this very quickly, solid lines are the development timeline for a product. If it moves into a dotted line, that's the point at which the organism was commercialized. If there's an arrowhead at the end of the line, it tells you that the product is still on the market. If there's a solid dot, it shows you that the product was taken off the market or was never commercialized in the first place. We can see here again very quickly kind of very different timelines of development, right? And we've got examples that go back to the early 90s. We've got ones that are much more recently being developed. So just trying to see what we can learn from a sort of comparison of these products. And then the approach that we took was, I think I said I was going to introduce this idea of social containment. So we thought about containment in two sort of complementary ways. The first one was really thinking about leaning into this idea of technological containment, asking what kinds of strategies have been used in these case study organisms to control and spread, or to control the spread of these organisms and their genetic material. So this is a form of containment that is really about maintaining some sort of separation between the organism and the world that it's in, right? So really trying to control and keep separate in some way. The other approach that we took was to really try and flip this almost and think about social containment more as a system of holding together. So thinking about the genetically engineered organism in the world and how the system is held together or stressed or taxed in some way through the introduction of the organism. So this is really trying to take that look at, say, actually containment is not just about it may not just be about keeping an organism separate, but maybe about thinking about how do you get the system around the organism to hold together. When we say the system, we kind of mean the world. We tried to think about a structured way of breaking this down. And so what we did with each case study was we took a sort of somewhat systematic dive into thinking through several different dimensions associated with that particular product. So we considered economic, spatial, temporal, ecological, cultural, public trust, regulatory, and ownership dimensions. These obviously overlap. It's not a complete picture, but we tried to sort of think through how these various dimensions independently influenced the trajectory of that particular organism through the development and commercialization pipeline, and also how they worked together to either enable the system around it or to constrain or threaten the viability of the system. In doing so, we were able to generate these sort of radar charts for each organism. I've just put 3 here just to kind of give you a sense of how it looks, kind of how it works, is that one thing you can quickly see is that each organism, so across the 11 case studies, they each have their own footprint where you can sort of see which dimensions prove to be the most challenging. So the way, the sort of, I guess the axis for this is thinking about how disruptive that particular element or dimension was. So if it's a 3, it was a highly disruptive element. If it's a 2, it was moderately disruptive. If it was a 1, it was little to no disruption. And you can begin to look at how clusters of variables come together in order to sort of enable an organism to come to market with very little challenge, or that sort of challenged its viability and progression through the development process. In this sense, the smaller the circle, the sort of smoother the pathway was. So I'll just give you 3 quick takeaways. I'm not going to dive into any specific case studies, but 3 kind of broad takeaways from the report. We looked at 11 case studies. Across these 11, what we found was actually very little correlation between technological containment strategies and successful social containment. If you're interested in digging into this, I might recommend that you read the Aqua Advantage salmon case and contrast it with the purple tomato case. So there's sort of 2 examples of that have quite different pathways. just to show you here, this is a very kind of busy slide that puts all 11 case studies side by side. That middle column shows you whether or not the developers built in any kind of technological containment strategies, physical or genetic containment strategies. And then the right-hand column shows you the current outcome. So was it commercialized? Is it not being commercialized? Was it pulled off the market? Is it still under development? Just draw your attention to the 4 products that we studied that are still on the market. None of them have any kind of intrinsic biocontainment built in. And indeed, among the case studies that we looked at, the products that did have some kind of built-in containment either were not commercialized or were taken off the market. So there's some food for thought here, perhaps for developers, in thinking through that relationship, right? I think this is obviously a limited sample. We only looked at 11 case studies. There's many GM crops out there that we didn't look at. But just to sort of challenge this default assumption that technological containment paves the way for regulatory approval, public acceptance, and so on, I think there's a lot here that signals that there's something deeper to unpack. Second takeaway, Richard, I think, mentioned this in his overview of the first kind of EMERS workshop. It's really helpful to know the likely regulatory path for your product as early as possible in the development process. So this is maybe a takeaway more for developers, but also sort of a conversation to be had with regulators. And this is partly, again, these are all sort of These are US-based case studies. This is grounded in the US context. But what we see here is that of the 11 case studies we looked, and I would suggest if you're interested in this, maybe look at the Oxitec mosquitoes case and contrast it with the Pivot Bio soil microbes case study. Again, two examples of how products have kind of made their way through the regulatory approval process very differently. Of the 11 case studies that we looked at, 9 of them have kind of engaged with the US regulatory system. What we see, again, building on Richard's comment, is most of them take very different pathways through the regulatory system. In some ways, this is by design, right? We have a coordinated framework in the US that assigns different jurisdictional authority to different types of products. Within an agency, there may also be different pathways depending on what your product is. And I think one thing is this, what this points to is a possible fragmentation of risk assessment expertise in the US. So I think, again, that the recommendation coming out of the first workshop report about having some sort of more centralized approach to guiding developers and to making some of these early decisions feels reasonably important based on this. I will say, too, so 2 of the cases that we looked at, the Oxitec mosquitoes and the Aqua Advantage salmon, really got snarled up in the regulatory system. And, again, just as we were trying to think about why that might be, what became clear was that these 2 particular products wasn't so much about the novelty of the product per se. You could argue that Pivot Bio's soil microbes are a very novel type of product, but it was the ambiguity around how the products should be classified within the regulatory system, right? So is the Oxitec mosquito an animal drug? Is it, is it something that should be evaluated by the FDA, by the EPA, by the USDA? And that lack of clarity about how to classify the organism, it led to significant regulatory delay in both of these cases. And I think as we see new types of products being developed and put out there, this is, again, a challenge for regulators to really understand and have early conversations about how will the product be classified. Because if you don't know that, it may end up getting bounced around within the US regulatory system. The third takeaway is, perhaps a fairly, not an earth-shattering one, but that public trust and stakeholder views on genetically engineered organisms are highly context-specific. And I think this is really important to emphasize because it's not uncommon to hear, at least within the scientific community, this sort of general malaise that, the public just really don't understand GM crops, so they're opposed to GM organisms. And I think what we saw through our case studies is that this is a much more nuanced issue. And again, I would encourage you, if you're interested in this, to maybe look specifically at the Terminator seeds case study and the Rainbow Papaya case study for kind of 2 very different sort of ways of both involving the public and then securing different levels of trust and acceptance in the product. Again, this is very heuristic, but if you just want to sort of put the cases that we looked at on a spectrum of sorts, we could say that there are a number that have to date raised very little public concern and ones that have been the source of much higher levels of public controversy. Just a couple of observations to make here is that the ability to choose or to opt in to using a product, engaging with a particular product seems very important across the cases that we looked at. And also to raise the notion that disruption is actually seems really interesting. It's not clear that the disruption of existing cultural practices is necessarily undesirable. So there were cases that we looked at where the disruption of cultural practices really did mobilize a lot of public opposition. So in the Terminator Seeds case, in the Aqua Advantage Salmon case. But in other instances, some disruption of the status quo aligned well with stakeholder values. So this is a case maybe too where we can look back at what Pivot Bio has been doing, working closely with farmers to understand their concerns around the stability of inputs, their fertilizer use, and so on, and actually saying that some disruption of their practices has been welcomed. So just to point to the fact that, making sort of blanket statements about how the public responds to GM crops is a very nuanced issue, and that I think, again, we can look to these case studies to get some insights as to what feels important here. Just to finish up with sort of a couple of additional emerging challenges that we're seeing, I think Richard pointed to this as well already, that across the case studies that we looked at, I think there are foundationally different logics of containment at play, including both the sort of farm and field type logics. This is something that we're actually trying to really work up in more detail now. Some of the organisms are ones that are really intended for sort of short-term, very spatially, temporally controlled deployment. They get planted and harvested at the end of a season. We also have organisms, for example, that are being designed both to persist in the environment, so genetically engineered chestnut tree, gene drives intending to spread in the environment. They're also intended to reconfigure ecological relationships, right? So it is not a case of just maintaining that separation between the organism and the world that it's in. It's actually intended to have an effect on the world. And I think for risk assessment purposes, this maybe raises questions around what does successful containment mean? What is acceptable persistence? And those kinds of questions that, I'm hopeful that we can maybe dig into. Richard also mentioned the sort of number of products that we're seeing that are maybe being marketed directly to consumers, and the kinds of challenges this might raise. On the one hand, really interesting to see this growing, increasingly lively marketplace for individuals choosing to purchase genetically engineered living organisms. It does also raise questions around cultural stewardship of these organisms, right? And these are things that we should maybe be attending to. So this is something actually that we have a recent policy briefing out on this if anybody is interested. So just to finish up to say, trying to introduce this kind of complementary way of thinking about containment, so really acknowledging both the sort of deep technological advances that we might be able to make in this space, but also flipping to the perspective of really thinking about social containment as how we hold the system together, where we have to engage with a much wider range of variables. This is really about how we enable and support a genetically engineered organism to be in the world, not necessarily to maintain its separation from the world. So I guess we're curious from the perspective of those of you who are working more from the development perspective to understand whether this kind of framework offers a useful set of questions and variables if you're considering deployment in open environments, really pushing us to think beyond safety and security, more towards thinking about the fit in or the fit with the world around it, right? If these are products that we're hoping will make a difference in the world, then centering what those variables are and how we sort of contain that system so that it can flourish is perhaps an important set of questions to grapple with. So this, just to thank the project teams, this is my group at ASU that wrote the sort of researched the case studies and put together the report. This project was also part of a bigger NSF-funded grant with collaborators at Rice and the University of New Mexico, who've been fabulous contributors and really helped us and kind of test some of these ideas. And there again is the link if you're interested in downloading the report. So thank you.